Hi, we're going to talk about large-scale patterns and how species and populations are distributed. So this is from chapter 9 in the book Ecology Concepts and Applications by Molles and Sher, published in 2019 by McGraw Hill. So this is the 8th edition of this textbook. And we've already talked about some of the uh, broader distribution limits on species. We've talked about small scale patterns and species distributions. Now we're going to talk about large scale patterns and species distributions, which is section 9.3 of the chapter. Then I'm going to go on to 9.4, which is on the size of the organism and how that relates to the density of its populations. Then we are also going to cover the applications section of the chapter, which is on the 7 forms of rarity and 1 of abundance that was developed by Deborah Rabinowitz. And then we shall finish up chapter 9. So it starts off talking about on large scales. When we look at, remember we had those patterns of how populations were distributed on small scales. They could be our uniform regular pattern, they could be a random pattern, or they could be a clump pattern. Well, we do find on large scales, individuals within a population are clumped. And it starts off talking about 2 large scale studies that are conducted every year on birds, the Christmas bird count and the North American breeding bird survey. The Christmas bird count is obviously done sometime in the winter. It's a week around the Christmas time, and that is done by amateur birders, but they use a common system to count the number of birds that they see that are resident for the winter time. And there's like thousands of localities, there's tens of thousands of people that participate in order to come up with this data. The North American Breeding Bird Survey is one that's done in the bird breeding season. So this is going to be done all across North America, but it's the opposite season from the Christmas Bird Survey. And what they usually do, it's usually done in June, and there's about 2,000 sites across the United States and Canada, and they all use the same methodology for doing point samples of the number of birds that they find in these different locations. To start with, Terry Root mapped out the patterns of bird abundance across North America from the Christmas bird counts data, and he found that on a continental scale, the bird populations showed clump distributions. So clump patterns occurred in species with widespread distributions, and he looked at a series of maps for 346 species at a continental scale and found clumped patterns for them. Jim Brown and colleagues did the same thing, but they were looking at the breeding bird survey, and they found that the breeding birds were also clumped. And the way they determined that was they saw that a small proportion of the study sites gave you the most numbers for each bird species in the breeding bird survey. So very few sites had really huge numbers, so they had much more clump distribution. So this is the data from the work by Root, and we're looking at the winter distribution on the top here of the American crow. And so we can see the American crow is found all across the US and up into eastern Canada. But when we look at the high populations of it, which are shown with the red color, we can see that there are certain areas where there's just very high concentrations of these crows during the winter time, and the rest of the places you find at much lower concentrations. So it's a very clumped distribution. The lower graph or the lower map is showing you the winter distribution of a fish crow. And fish crows are only found in a very restricted area, which is along the coast of the Gulf of Mexico and the southern Atlantic coast of the US. And we can see that there's just a couple of hot spots, one in the Mississippi Delta, one in west of Tallahassee, Florida, and a third in central Florida. So this bird population is obviously just clumped in a few very good habitats where the species do extremely well. we look at the type of data that the Browns did with the breeding bird survey. Here's an example. This is looking at the red-eyed vireo, which is a small songbird. And we can see on the x-axis we have for each point where they were counting birds on a route. So the breeding bird survey is done along these routes where they stop every half mile and count the number of birds. So along the routes on the x-axis, we have how many vireos we found on this route and how many vireos we found in that route. The y-axis is how many routes we found with that number. So we can see this point right here, we found most of the routes had very small numbers of vireos. And if we look at the number of routes that say had 100 vireos, there's only one route has 100 vireos. One route has 148 vireos, but then over 200 routes found one vireo. So we can find very few vireos most of the time, and then you just found these few spaces along the right side of the graph, these few routes where you had the highest concentration of your vireos. So basically they're proposing that these distributions are clumped because the environment varies, and the individuals are going to be found in higher numbers where the environment is favorable for those species. Now, a different approach was taken around the 1950s by Robert Whitaker. And he was looking at, instead of looking at all of North America, he thought he could look at a gradient in environments in a very much shorter distance, and that's by going up a mountainside. So you're going up a moisture gradient as you're going up the sides of a mountain range, and you can go from sort of something that's hot and dry at the bottom to cooler and wetter towards the top as it cools off. So Whitaker was collecting data at various spots, and you know quite a bit about mountains now because we've talked about them with biomes. There's quite a variation in environments as you go up a mountainside. And so he did one study that he was looking at the gradient in Arizona, where he's going from he's in the Santa Catalina Mountains in southern Arizona, and you're out there in the Sonoran Desert, but you look up the mountains, and the mountains are all green. They're covered in nice green vegetation. So as you go up the mountainside, there's a moisture gradient, and it depends on what facing slope you are on. So if you're actually on a south facing slope that's getting blasted by the sun, you might find a very different gradient than if you're on the north facing slope. So he's going up this slope from a fairly moist canyon at the bottom up across a southwest facing slope that's sort of getting beaten by the sun, and then up to the very tall tops of the slope, and looking at the distribution of the trees or the plants as it's going up. What he found was a clump distribution along the moisture gradient, where different species were clumped in the environment, were found in the highest densities in the environments that they were best suited for, and then you'd find less of them as you moved further up or down. So here we can see this is a proportion of the different tree species across the x-axis, and on the y-axis we're going uphill from the lower slopes to the middle slopes to the high slopes. And the Douglas fir trees are really just found down here where it is moist down in the canyon, so it's a little bit shaded more. And then as we're going up slope, you start to find more of the madrones until they peak, and then you go up higher and they decline. And then we go up even higher and we find the pinyon pines. And at first you don't find so many in the middle slopes, and then as you get to the higher and higher slopes, you get more and more of these pinyon pines. So you get these kind of bell-shaped distributions, if you turn this on the side, where you find them concentrated in the areas where they are most suitably adapted. He didn't just do this in the desert, he decided he would do this in different places. Whitaker did this in several places. He also did this in the Great Smoky Mountains in eastern North America, where you have a moisture gradient from a moist valley bottom to a dry south-facing slope, southwest-facing slope again. And here he could find that we've got three species plotted on here. We have the hemlocks that are found down in the valley bottoms. Then you find the red maples. They're very low numbers in the valley bottoms, but on the mid slopes they're doing well. But then when it gets up higher, they don't do as well. And then at the very tops, you start to get, you can see the maples going up to the pinyon pines in the photograph here. You can see the sort of, not the pinyon pines, the Table Mountain pines at the tops of the slopes. And so once again, even though these species will overlap each other along the gradient, they're found in lower numbers as you get away from the conditions where they are doing the best. So just like the birds, the tree populations are concentrated in hot spots. Next, we're going to be looking at population density and see how that's influenced by organism size. But you should be able to answer these concept review questions at the end of section 9.3. So even though we've been talking about the different types of population densities in certain areas being clumped or being uniform, if the population's spread out or concentrated, there is one other factor that is affecting the population density that we haven't talked about, and that's the size of the organism. And we know in general that the population density declines with bigger organisms. So if you have little tiny things, you can pack more into a place. They use up less resources. So you might have, billions of bacteria in the same in a very small place, but at the same time you might have a large mammal or bird like an eagle. You might have just one eagle for every square kilometer. So taking a look at how the size of the organism is impacting the density of the organism is really interesting. The work by Damath found that the population density he looked at was with herbivorous mammals, and he was looking at basically how the body size and population density were related. And then this was built on by looking at other animals. So Peters and Wassenberg looked at aquatic invertebrates and found something similar. And let's take a look at the data here. So we've got the body mass on the x-axis, and then on the y-axis we have the density of the herbivores. And this is the work that was done in the 1980s by Damath. And we can see, even though there's quite a bit of scatter, there's a lot of points on this graph. We can definitely see there's a clear trend line as we are going from the smaller herbivorous mammals to the larger herbivorous mammals. We can see that the density of the populations of the large mammals is going to be much lower than the density of the populations of the small mammals who have much higher population densities. This is from the sort of expanded work that was done by Peterson and Wassenberg after Damath. And this one, once again, we have the same axes. We've got on the x-axis, we've got the body mass of the animals, and on the y-axis, we have the density. And we can see, once again, we have a very clear trend line. It's really cool that the bigger animals, no matter what kind, the bigger animals are found in lower densities than the smaller animals are found in higher densities. I think this might be related to energetics. It seems like a pretty clear pattern. They've also outlined for you that the different types of animals we're looking at. So here in red, we've got the aquatic invertebrates, which tend to be in higher density than the terrestrial vertebrates, which are in this light blue. So where you can see that they do overlap a certain amount, but there's a lot of space here where the aquatic invertebrates have higher densities than some of these terrestrial vertebrates, but it depends on how big they are and where they are. So there is some overlap between these groups. This one in orange down here, the birds, seems to be at lower densities than we are finding for the mammals, which are above them, and then the vertebrate poikilotherms, and remember we talked about what poikilotherms were. So the poikilotherms, the warm-blooded animals, seem to be in higher densities as well. So you find some particularly interesting patterns showing up here. And so you see, in general, we see this overall trend, but aquatic invertebrates tend to have higher densities than terrestrial invertebrates the same size. Mammals tend to have higher population densities than birds of similar sizes. And so it makes sense to look at each of these groups individually. That's animals. Let's talk about plants. Plant size and population density. So we also know the same thing with plants. Plant population density decreases with increasing plant size. But the reasons for this seem to be quite different. Tree seedlings can live at very high densities, but as the trees grow, the density declines until the mature trees are at low densities. This is some work that was done over a wide range of plant sizes and plant types by Jim White, and we're looking at things from as small as duckweed. Duckweed is a little tiny flowering plant, grows on the surface of your ponds in the summertime. Little tiny thing. It just has a couple of little leaves. You'll see it floating on the water with a couple of little leaves and a little root hanging down on the surface of the water. So it's very, very tiny, but it can live at very, very high densities. And then at the other extreme, we have the sequoia, the coastal redwood trees. So on that end, we've got the redwoods. So they are found very big, very big trees, but they've got very low densities. You don't find very many at a time. So we can see a very, very clear line here as we go from bigger plants have low densities to smaller plants have very high densities. And so we're going from the trees to clonal perennial herbs. So these are things like grasses, things that die back every year. They're not woody like the trees and shrubs. And then these are the annual herbs, which are even smaller. And they have to set seed every year and come back, sprout back from the seeds. Okay, you're probably going to want to take the time to draw this table as we go. And I'm just going to give myself a little template to start with. So we're going to deal with 7 forms of rarity and 1 form of commonness. So when you're looking at this table, you're going to have 8 boxes. So I'm going to just make a table with my 8 boxes. Divide those up here and here, here and here. So the first factor we're going to talk about is geographic range of the species. So I'm going to use that top row that I've got here for talking about geographic range. So I'm just going to write that. Geographic range. And I'm basically going to divide this table, and obviously it's a continuum, but we're going to divide this table into two sides. And one side of the table, the 4 boxes on the left, will have the species that have a very broad geographic range. So it's found in a lot of different places. So I think they call that extensive in your textbook. So we'll call that extensive versus a narrow or restricted range. So half of these are already going to be rare in one sense because they are restricted in their range. And then we're going to look at how many different types of habitats they can tolerate. So that would be their habitat tolerance. So the next row down then, we're going to call habitat tolerance. Habitat tolerance. And we're going to have 2 categories for that. We're going to have the species that can live in a broad range of conditions versus the species that have very specific narrow habitat requirements. So each one of these sides ends up being divided into 2 tolerance boxes. We've got broad, and narrow tolerance. And here, broad and narrow tolerance. So this first box, we have species that have an extensive range and a broad number of habitats. So those ones are not limited at all. Anywhere else you're limited at least in some way. You either have limited by your habitats so that you're going to be in the narrow category, or you're going to be restricted geographically on the right side of the graph. And then obviously the ones that you find the least are going to be restricted geographically, and they're going to be in a very narrow range of habitats. So we have one more factor to deal with, and that is when you find the species, what is its population size? So that's going to be going down here. We're going to be looking at the population size when you find it. And we're just going to have that large versus small. So we'll have just large and small. So now we have this first top left box where you have species that have an extensive range, a broad number of habitats can live with, and you find it in a large number, in large populations when you find it. Those are not rare species. Those are your common species. Everybody else, every other box is rare in some way. So anything on the bottom row is rare because it has a small number of individuals when you find it. Anything in the narrow habitat columns are restricted to the habitats they're found in. And everything on the right side of the diagram is going to be restricted on its geographic range, just found in very few places. It might be found on the island of Madagascar. Restricted range might be found only in old growth tropical rainforests, and when you find it, it has a small population. That box right there, those are where you're going to find a lot of species that are in danger of extinction. Something like your lemurs in the tropical rainforests in Madagascar with small populations. So here's a nicer looking picture with some examples of some species in them, and set up the same way. You have a large or small geographic range at the top. So there's a geographic range. You've got the habitat specificity. Then you have your population size when you do find them. And we've got the species that we see commonly right here in the first box, and everything else in yellow is rare in some way. So your textbook shows you some examples, shows you going sort of from the most common species to the rarest species. Obviously, we know more about the common species than we do the rare species. This system is widely used. This has been found to be a really useful, even though it's like these are very simple, broad categories. It's really helped us come up with setting priorities on what species we're going to concentrate on protecting. So this is the way that they've sort of portrayed that in the book, going from the most common species at the top, which have an extensive geographic range, broad habitat tolerance, and large local populations, down to your most vulnerable species at the bottom, which have a restricted geographic range, narrow habitat tolerance, and a small local population. And they've given you some examples of both animals and plants and in these categories. In the United States, this sorts of classification of species and of ecosystems, of communities, has been done in every state, and we have called it the Heritage Inventory System. This was originally set up by the Nature Conservancy, which was set up by a group of ecologists, and they wanted to find out where the most vulnerable species were, so they needed some sort of ranking system. And so the Heritage Inventory System is based on these three criteria, and they're able to rank species on their rarity within a state, but also their rarity nationally and their rarity globally. And so they can put them then into different categories depending on these rankings. So things with the lower number are the more endangered species, the things that are more difficult to find and they need our help. So you can see there's global ranks G1, national ranks N1, state ranks S1, and these are the criteria that they use to do that. And that way you can concentrate on the species that need our help the most, and know which ones are secure and widespread, and then which ones that are obviously going to need more of our help. So throughout this chapter, we've talked about the first presentation was on distribution limits. Remember, we had the kangaroos in that one. We talked about distribution patterns, clumped patterns, regular patterns, and random patterns. And we talked about the relationship between big organisms have lower population densities, and lastly talked about commonness and rarity. So go back through the chapter. You should be able to answer the concept review questions 1 and 2 for section 1, questions 2 and 3 in section 2, questions 1, 2, and 3 in section 3. Don't worry about the question in section 4. And then at the end of your chapter, there's chapter review questions 1, 3, 5, 6, 7, 8. So make sure you can answer all of those. Those will help you on your exam. And then go to the end of the textbook. There's an Investigating the Evidence, section 9, which is on clumped, random, and regular distributions, which actually shows you how to measure these distributions and be able to answer the first question, which is dealing with those creosote bushes we talked about when we were dealing with that concept in section 9.2. Following this, we're going to move on to the next chapter. Next two chapters are dealing with populations, population dynamics, and population growth. And we're going to start off with looking at what influences how populations change in number, and then how we model that with population growth. So we will see you soon. See you there.